So when you hear the word steroids, what instantly pops into your head?
Usually it's a pretty binary thing for most people.
Right. I mean, you're probably picturing like illicit muscle building drugs for sports, or maybe you're just thinking of that basic over-the-counter hydrocortisone cream you put on a bug bite.
Exactly.
But what's actually happening inside your body with these chemicals is, honestly, it's so much stranger and far more miraculous than either of those two extremes.
It really is. And the confusion, um, it makes total sense because we use that single word steroids as this massive umbrella term.
Yeah.
We use it to describe all these compounds that share a basic foundational chemical structure. It's essentially this microscopic skeleton made of carbon rings.
Right.
But depending on how that skeleton is decorated with different molecules, these compounds perform wildly different literal life or death jobs inside you.
And unpacking those life or death jobs is our mission for this deep dive. We're taking a stack of pharmacological research and some pretty wild medical history to explore the fascinating world of glucocorticoids versus mineralocorticoids.
It's a mouthful, but it's crucial stuff.
Totally. We're going to look at these two distinct steroid effects, how they literally masquerade as one another inside your cells, and why a century-old medical discovery actually became a frontline therapy in a modern pandemic.
Which is just an incredible story on its own.
It is, but to start, we really need to look at a system that governs almost everything, keeping you, the listener, alive today, right?
Yeah, because understanding these specific hormones is essentially the key to understanding how human biology manages everything from, say, an acute panic attack to systemic inflammation to something as basic and mechanical as the blood pressure in your veins.
Wow.
If you don't grasp how these two specific classes of steroids function, modern medicine can look like magic, rather than the intricate engineering it actually is.
So to understand that engineering, we have to visit the manufacturing floor. We have to go down to the adrenal glands,
Right.
which are these two small kind of triangular organs sitting right on top of your kidneys. And the outer layer of these glands, the adrenal cortex, it operates like a highly compartmentalized pharmaceutical factory.
The strict anatomical division of labor there it's just incredible.
Yeah.
Yeah, the adrenal cortex is structured in 3 concentric microscopic zones. Like, medical students have used this mnemonic for generations to remember them from the outside in.
The GFR one.
Exactly, GFR.
Yeah.
The zona glomerulosa, the zona fasciculata, and the zona reticularis. And you pair that GFR with the phrase salt, sugar, sex to remember what each specific layer is producing.
Salt, sugar, sex. It's definitely memorable.
It works.
Let's look at that middle layer, though, the sugar layer. The zona fasciculata.
Okay.
That's where glucocorticoids are manufactured. And in humans, the main product rolling off this assembly line is cortisol, right?
That's the one, your ultimate stress hormone.
Right, so it regulates glucose metabolism, which, that's where we get the
Yeah.
glucocorticoid. And it suppresses and just fundamentally reshapes your immune and inflammatory responses.
Yeah.
And the boss telling this assembly line what to do is the HPA axis, which is basically your brain, right?
Yeah, specifically your hypothalamus and pituitary gland.
Sounding an alarm and telling your adrenal gland, hey, we are under stress, flood the system with cortisol.
That's exactly it. But then moving to the outermost layer, you have the salt layer, the zona glomerulosa.
Okay, the salt factory.
Right. This layer produces mineralocorticoids, primarily a hormone called aldosterone.
Aldosterone, okay.
And its main job is managing electrolytes, fluid balance, and essentially dictating your total blood volume and blood pressure.
How does it do that?
It gives your kidneys a very, very specific instruction. It says, hold on to sodium and get rid of potassium.
Oh, I see.
And because where water goes, salt goes. Retaining salt retains water, which keeps your blood volume up and therefore your blood pressure up.
But the boss for this salt factory isn't the brain, right?
No, the brain isn't involved here at all. The boss of the salt factory is the RAAS system.
RAAS.
Yeah, RAAS, the renin-angiotensin-aldosterone system. You can think of RAAS like an internal plumbing sensor.
Oh, plumbing sensor. I like that.
Yeah. If the specialized cells in your kidneys detect that the blood pressure in the pipes is getting too low, they release this enzyme called renin.
Okay.
Renin acts like an alarm bell in the blood, triggering a whole cascade that produces angiotensin, which is a really potent constrictor of blood vessels.
Right.
Angiotensin then goes, knocks on the door of the adrenal gland and says, release aldosterone. So you have the brain managing stress and sugar in the middle layer. and this highly localized plumbing system managing salt and pressure in the outer layer.
So we have this one factory using the exact same raw material cholesterol for both of these assembly lines.
Exactly.
But they have completely different bosses and totally different agendas. The brain is shouting at the middle layer to manage stress, and the kidneys are shouting at the outer layer to manage pressure.
It's a crowded factory.
Right. So if they're packed so tightly together using the same ingredients, do these two pathways ever, accidentally interfere with each other?
Oh, they interfere in profoundly consequential ways.
Really?
Yeah, but to really grasp why they cross wires and why this neat, tidy textbook division of salt versus sugar is actually dangerously incomplete, we have to look at how these hormones were discovered.
Okay, let's get into the history.
The historical context reveals why we had such massive blind spots for so long.
The history here is fascinating, though it starts out pretty grim, honestly.
Very grim.
Back in 1855, this British physician named Thomas Addison began describing this fatal, mysterious wasting disease.
Addison's disease.
Right.
Yeah.
His patients were developing this odd, dark pigmentation on their skin, losing massive amounts of weight and eventually dying. And he performed autopsies and realized their adrenal glands were completely destroyed.
Which was a huge revelation at the time.
Yeah, because it proved the adrenal cortex was absolutely essential for life. But in 1855, they had literally no idea what the gland was actually producing.
Right. They just knew that without whatever was in that tissue, you died.
Right.
And that realization kicked off this massive international chemical isolation race, which really peaked a lot later in the 1930s and 1940s.
Half a century later.
Yeah. Researchers like Edward Kendall at the Mayo Clinic and Thaddeus Reichstein in Switzerland, they were painstakingly grinding up tons, literal tons of animal adrenal glands, trying to isolate these tiny crystalline compounds.
This blind trial and error.
Completely. They didn't know what they were doing chemically, so they were just labeling them alphabetically. Compound A, compound B, compound E, and so on.
And that painstaking chemistry kind of sets the stage for what has to be one of the most cinematic moments in modern medical history.
Yeah, absolutely.
So it's September 21st, 1948. A rheumatologist named Philip Hench at the Mayo Clinic decides to take compound E, which we now know today as cortisone,
Right.
and he injects it into a 29-year-old woman named Mrs. Gardner. She had been bedridden for years, completely immobilized by severe agonizer rheumatoid arthritis. Her joints were just locked up with inflammation.
And the historical accounts of what happened next, they read like pure fiction.
Right.
Within just a few days of receiving this completely novel compound, Mrs. Gardner isn't just reporting that her pain is a little better. She's out of bed.
It's crazy.
She's walking down the hospital corridors normally. She literally leaves the hospital and goes shopping downtown.
Just a few days later.
The sheer power of this molecule to just turn off severe systemic inflammation was unlike anything medicine had ever seen.
I try to imagine being a doctor in the room witnessing that. It was such a theatrical, instantaneous reversal of chronic disease.
It changed everything.
And by 1950, just 2 years after that injection, Kendall, Reichstein, and Hench are standing on a stage in Stockholm sharing the Nobel Prize.
Unheard of speed for a Nobel.
Yeah. The glucocorticoid is introduced to the world as this absolute therapeutic miracle. But that was just the sugar side of the factory. What about the salt side?
Right, well, the salt side was isolated a bit later in 1953 and to much less public fanfare.
Why is that?
Researchers had noticed something peculiar in the lab. Animals that had their adrenal glands removed were mostly dying from massive salt wasting and dehydration, not from a lack of blood sugar.
Oh, wow.
Their bodies just couldn't hold on to water. So researchers Sylvia Simpson and James Tate in London managed to isolate this separate, highly potent salt-regulating substance from the adrenal extract.
And that was the mineralocorticoid.
Exactly. They initially called it electrocortin before eventually settling on the name aldosterone.
Okay, so by the mid-1950s, the medical community has their sugar hormone, cortisol, which performs absolute miracles for inflammation.
Yeah.
And they have their salt hormone, aldosterone, which keeps patients from dying of dehydration.
Right.
But I'm wondering, if cortisone was such a precise miracle drug for rheumatoid arthritis, did scientists back then just assume these hormones operated like perfectly cut keys that only fit into one specific lock in the body?
That was exactly the prevailing paradigm for decades. Yeah, the scientific community embraced this strict 2 hormones, 2 locks model. The assumption was that cortisol travels through the blood and only goes to the glucocorticoid receptor, turning on the sugar and stress pathways.
Okay.
And aldosterone travels through the blood and only goes to the mineralocorticoid receptor, turning on the salt pathways. It was a simple, clean, elegant way to understand human biology.
But biology rarely gives us simple and clean.
No, it does not. The truth didn't emerge until the 1980s, and when it did, it absolutely broke all the established rules of how hormone receptors were supposed to function.
Let's look at that 1980s molecular twist, because this is where it gets wild.
Really does.
Molecular biologist Ronald Evans manages to actually clone the human glucocorticoid receptor in 1985. 2 years later, in 1987, his team clones the mineralocorticoid receptor.
Right.
But when they finally looked at the underlying genetics and the three-dimensional molecular structure of these receptors, they uncovered what became known as the promiscuity problem.
Yes. The sequencing data presented this massive biological paradox.
What did it show?
It showed that the mineralocorticoid receptor, the lock that was supposed to be exclusively designed for the salt hormone, aldosterone wasn't picky at all.
Not at all.
Its steroid-binding pocket actually bound cortisol just as tightly and with just as much affinity as it bound aldosterone.
And to put into perspective why that is so terrifying for human biology, cortisol is floating around in your bloodstream at roughly 1,000 times the concentration of aldosterone.
The math of that is just staggering.
Right.
Think about it. If cortisol binds to the salt receptor perfectly, and there is 1,000 times more cortisol circulating in the blood, then cortisol should simply drown out the aldosterone.
It should just take over.
It should. It should constantly flood your kidneys, permanently activate all those salt receptors, drive your blood pressure through the ceiling, and kill you.
But it doesn't.
It doesn't. And resolving why we aren't all dying of high blood pressure right now required a massive conceptual leap.
I want to try to visualize this molecular chaos.
Okay, let's hear it.
It sounds like a nightclub.
A nightclub.
Yeah. You have one guy named aldosterone trying to get in, and 1,000 guys named cortisol trying to rush the door at the exact same time.
Okay.
The receptor is the door, and the door itself doesn't care who walks through. But there's a specialized enzyme in the kidney called 11-beta-HSD2.
Yep.
And that enzyme is basically the VIP bouncer standing outside, violently tossing all the cortisols out into the alley, deactivating them so only aldosterone can actually reach the door. Is that how he survived this?
That analogy captures the mechanics perfectly.
Really?
Yes, the bouncer protected the door. The scientific term for this is pre-receptor selectivity.
Pre-receptor, meaning before the lock.
Exactly. It means the receptor isn't what discriminates between the two hormones, the local tissue discriminates. That 11-beta-HSD2 enzyme acts as a localized gatekeeper in the kidney.
Wow.
Before cortisol can ever reach the receptor, the enzyme grabs it, alters its chemical structure, and turns it into an inactive byproduct called cortisone. Yeah. This shift from thinking the lock was picky to realizing there's a molecular bouncer out front fundamentally changed endocrinology.
And we know this isn't just a theory, right? We can see what happens when the bouncer goes missing.
Sadly, yes. We see the devastating consequences in a rare genetic disease called apparent mineralocorticoid excess, or AME.
AME.
A person born with AME is missing the genetic instructions to build that bouncer enzyme.
Oh, no.
So without it, the cortisol rushes the door. It completely floods the kidney's mineralocorticoid receptors. The patient develops severe, life-threatening hypertension and dangerously low potassium levels at a very young age.
That's awful.
It is. Clinically, it looks exactly like their body is overproducing massive amounts of aldosterone. But blood tests show their actual aldosterone levels are practically zero.
Wait, zero?
Yeah. The body's RAAS plumbing system is frantically trying to shut the salt retention down, but it can't, because cortisol has completely hijacked the machinery.
It is terrifying to think our survival hinges on one exhausted enzyme working overtime in our kidneys.
It really puts things in perspective.
And if you're listening to this, you might actually have a real-world demonstration of this mechanism sitting in your pantry right now. Let's talk about the licorice experiment.
This is one of the cleanest and, frankly, most surprising natural demonstrations of this entire pharmacological pathway.
Right. We're talking about true black licorice here, not the red plastic-tasting cherry vines, but candy made from actual licorice root.
Huge distinction.
The root contains a remarkably sweet compound called glyceric acid. And it turns out glyceric acid is essentially a chemical sniper that takes out our nightclub bouncer.
It's incredible.
It actively binds to and inhibits that 11-beta-HSD2 enzyme.
Right. So if you consume enough true black licorice, You artificially induce a pharmacological version of that rare genetic disease we just discussed.
You give yourself AME.
Temporarily, yes. Without the enzyme functioning, the cortisol in your blood is no longer being deactivated. It floods those renal receptors. Your kidneys start aggressively hoarding salt and dumping potassium.
And your blood pressure spikes.
Exactly. It is a fully documented clinical entity called licorice-induced pseudohyperaldosteronism.
That is a very long name for candy poisoning.
It is. But there are actual peer-reviewed case reports of individuals eating large quantities of black licorice candy over a few weeks and ending up in the emergency room with fatal or near-fatal cardiac arrhythmias.
Just because their potassium dropped to critically low levels?
Yes, from candy.
We can accidentally break this incredibly complex life-or-death hormonal system with candy. But understanding the bouncer's limits also explains a lot of what we see in modern medical treatments, doesn't it?
It absolutely does.
Like, if you know someone who has been prescribed high doses of prednisone, which is a synthetic glucocorticoid for a long time to treat an autoimmune issue,
Very common.
they often develop high blood pressure and a rounded face, sometimes called a moon face, from water retention. That happens because the massive dose of the synthetic drug simply overwhelms the bouncer's capacity to deactivate it, right?
Exactly. The enzyme can only work so fast. You give someone a massive therapeutic dose of a glucocorticoid, and the enzyme simply can't process it all.
It spills over.
The drug spills over, hits the mineralocorticoid receptors in the kidney, and suddenly a medication you prescribed specifically to suppress inflammation is causing severe water retention and hypertension.
So if we can accidentally break this system with candy or, overwhelm it with arthritis medications, how do doctors intentionally manipulate these dual pathways to keep people alive?
The clearest example of managing both pathways is how we treat Addison's disease today.
Going back to Thomas Addison's patients in 1855.
Right, the ones who had lost their entire adrenal cortex. To keep a patient with Addison's alive today, modern medicine has to explicitly respect the two-axis model.
You can't just give them one pill.
No, you cannot just give them a glucocorticoid to replace the cortisol. You have to give them hydrocortisone to replace the stress and sugar hormone, and you have to prescribe a separate drug, fludrocortisone, which is a synthetic mineralocorticoid, to replace the salt and blood pressure hormone.
So if you try to cut corners and only replace one, the whole system crashes.
Instantly.
Right.
Replace only the cortisol, and the patient will still die from a massive salt-wasting crisis and cardiovascular collapse.
Wow.
Replace early the aldosterone, and they might maintain their blood pressure, but they will have zero stress tolerance. Their blood sugar would plummet, and a simple respiratory infection could be fatal because their body cannot mount a metabolic stress response.
The two systems are deeply distinct in their vital functions, even though they share the exact same locks at the molecular level.
That's the paradox we've been navigating.
Okay, so if this bouncer enzyme is so critical in protecting the kidneys, what happens in organs that don't have a bouncer?
That is a million-dollar question.
Like, what happens when these hormones hit the heart? Because for the longest time, the medical dogma was mineralocorticoids only act on the kidney, right? Aldosterone equals kidney equals blood pressure. End of story.
That dogma was deeply entrenched for decades. But researchers eventually asked that exact question. What happens if mineralocorticoid receptors exist in tissues outside the kidney? Tissues that lack the protective 11-beta-HSD2 enzyme.
And what did they find?
Well, in 1999, a massive landmark cardiology study called the USMLE trial investigated this. They took patients suffering from severe heart failure and gave them a drug called spironolactone.
Spironolactone, that's an MR antagonist, right? It physically blocks the mineralocorticoid receptor.
Exactly.
But up until that point, doctors basically viewed spironolactone as this weak, relatively minor diuretic, like a simple water pill to help you pee out a little extra fluid without losing too much potassium.
That's it. The cardiology world viewed it as a minor symptomatic treatment.
So,
But the results of the Rayleigh trial shocked everyone.
what happened?
They found a roughly 30% reduction in mortality among these severe heart failure patients.
30%.
A 30% survival benefit in heart failure is astronomical. And the data clearly showed it was not happening just because the drug was making them urinate more fluid.
So what was the drug actually doing to the heart muscle?
It proved that aldosterone isn't just a kidney hormone regulating fluid. Aldosterone has a direct, deeply damaging physiological effect on the heart tissue itself.
Really?
Yeah. When the heart is failing, the body's RAAS system panics and pumps out massive amounts of aldosterone. This aldosterone hits unprotected receptors in the heart and actively drives fibrosis.
Fibrosis, that's the creation of dense scar tissue, right?
Exactly. It causes the heart muscle to remodel in a rigid, maladaptive way, making it even harder for it to pump.
Oh, wow.
By giving spironolactone to block that receptor, cardiologists weren't just tweaking the patient's blood pressure, they were stopping the hormone from actively scarring the heart to death.
That completely revolutionized our understanding of cardiovascular disease.
It fundamentally changed how we treat heart failure.
So uncovering that cardiac fibrosis mechanism is a huge win for medical science. But I want to look at the flip side of this pharmacology, where the industry spent the 1990s and 2000s chasing what turned out to be a multi-billion-dollar ghost.
Ah, yes, the dissociated glucocorticoid.
Right. The pharmaceutical companies basically looked at cortisol and said, this stuff is a miracle for inflammation, but if you take it long-term, you gain weight, your bones thin out, you can develop diabetes, and your muscles waste away.
And the metabolic side effects of long-term steroid use are truly devastating for patients.
They are. But I'm stuck on something here, though. If we mapped the human genome and we know exactly what these molecular locks look like, why couldn't pharmaceutical companies just build a drug that hits the inflammation pathway but completely ignores the metabolism pathway?
It makes sense on paper.
Right. With billions of dollars in R&D, shouldn't untangling those two things just be a solvable chemistry problem?
It seems like it should be, but it failed because our conceptual model of how DNA works was far too simplistic.
How so?
We used to think of the glucocorticoid receptor acting on DNA like a control panel with two completely separate levers you could pull independently, one for inflammation, one for metabolism.
Okay.
But as molecular biology advanced, we realized those programs are intensely physically intertwined at the genetic level.
So they aren't separate levers on a control panel?
Not at all. Think of the genome not as a flat string of code, but as a densely packed, three-dimensional ball of origami.
Origami, okay.
When a steroid receptor binds to the DNA to turn off an inflammatory gene, the physical shifting and folding of that DNA bundle inevitably exposes or alters the adjacent genes responsible for fat storage or bone density.
Oh, I see.
The mechanism that silences inflammation is fundamentally mechanically tethered to the mechanism that alters glucose metabolism. You cannot pull the string to hide the inflammatory crease without simultaneously tightening the string on the metabolic side.
That's a great way to picture it.
And to this day, despite decades of effort and billions of dollars, no truly separated or dissociated, glucocorticoid has achieved clinical success.
The immense complexity of how these steroids interact with our immune system, it brings us to our final, incredibly relevant point. We have to talk about the recovery trial.
Yes, we do.
We are talking 2020, 2021, right in the darkest, most terrifying heights of the COVID-19 pandemic.
The recovery trial was a watershed moment in evidence-based medicine. It was a massive randomized platform trial run out of the UK, urgently evaluating potential treatments for COVID-19.
And one of the treatments they tested was dexamethasone.
Right, which is a highly potent, widely available, and very cheap synthetic glucocorticoid.
And the results of that trial perfectly highlight the absolute golden rule of these hormones. Pharmacological context is everything.
It really is.
Because the trial found that a low dose of dexamethasone significantly reduced mortality in COVID patients who were on ventilators or required supplemental oxygen.
A huge breakthrough.
But, and this is a massive distinction, it showed zero benefit and actually showed signs of active harm in patients who had mild COVID and did not need oxygen support.
And if we break down the timeline of a viral infection, that data makes perfect sense.
Why is that?
Glucocorticoids, by their very nature, suppress the immune system. Early on in a viral infection, your body's primary objective is viral clearance.
Right. Fighting the virus.
Exactly.
Right.
You need your immune system operating at full capacity to hunt down and destroy the replicating virus. If a doctor gives you a powerful steroid reflexively during that early phase just because you feel sick, they are chemically crippling your body's ability to fight off the pathogen.
That's where the harm comes from.
Yes.
But later on, in severe COVID cases, the virus replicating isn't actually the main threat anymore, is it?
No. In severe, late-stage COVID-19, the primary threat is the patient's own immune system overreacting.
The cytokine storm.
Exactly. It triggers a massive, uncontrolled inflammatory response that essentially begins destroying the patient's own lung tissue.
So at that specific critical moment, hitting the brakes with a potent glucocorticoid is life-saving.
Yes. It proved that a century-old class of hormones, the very same physiological concept discovered when Mrs. Gardner got out of her bed in 1948, could be repurposed as frontline pandemic therapy.
Amazing.
But it only worked because we finally understood the nuanced timing and context of the immune response.
Context is everything. So just to recap this incredible pharmacological journey for you listening, we started with the neat, tidy textbook division of the stress hormone from the middle layer and the salt hormone from the outer layer of the adrenal gland.
The factory.
The factory, right. We saw the miraculous clinical birth of cortisone in 1948. We uncovered the terrifying molecular twist of the 1980s, the shared lock, and the VIP bouncer enzyme, 11-beta-HSD2, desperately working to protect your kidneys from a lethal flood of cortisol.
The promiscuity problem.
Yep. We learned that the chemical compounds in black licorice can snipe that bouncer. And finally, we saw how blocking these specific pathways saves failing hearts, while understanding their delicate timing saves lungs.
The journey really highlights how dynamic and hidden pharmacology can be.
Absolutely.
And, I want to leave you with a final thought to mull over, building directly on that licorice revelation.
Okay, let's hear it.
We now know, as a scientific fact, that a simple sweet compound naturally found in a plant root can completely paralyze one of our body's most critical enzymatic bouncers, tricking our kidneys into a state of severe, life-threatening hypertension.
Right.
So it forces us to ask, how many other unrecognized, unstudied compounds floating around in our modern diet or our synthetic environment might be secretly flipping the microscopic switches of our endocrine system, turning our own life-saving hormones against us without us ever even realizing it?